In this section, we are going to discuss the physiology of receptors. This is a very important part in sensory physiology. So let us start with the definition of receptors. So what are receptors? Receptors can be defined as biological transducers. So we already know the meaning of biological. So what do we mean by transducer? You must have studied it in physics as well. Transducer is it's a device which converts one form of energy into another form. So receptors, they act as biological transducers. So let us try to understand it with the help of certain examples. For example, if we take a look at our eyes, in the retina of eyes, receptors for light are present. So these receptors, they are called as rods and cones. Rods and cones are the visual receptors present in the retina. And how do they act as transducers? They convert light energy into electrical energy. So once light falls on them, they generate impulses which go to higher centers. So those impulses, they are a kind of electrical energy. So light energy is converted to electrical energy. Similarly, in case of hearing, the hair cells act as receptors. Hair cells which are present in the inner ear, they act as receptors. And how do they act as transducers? Again, they convert sound energy or the energy from vibrations into electrical energy, into electrical impulses. So this is the definition of receptors. That is, receptors are biological transducers. Rods and cones and hair cells, they convert sound to electrical energy. Now, there are so many receptors in our body. So in order to have better understanding of receptors, there are some terms that we should be aware of first. Okay, so threshold stimulus, you must have already studied in nerve physiology. Threshold stimulus is the stimulus, minimum amount of stimulus, which takes a neuron or a nerve fiber from resting membrane potential to threshold to firing level. So once it reaches firing level, then action potential will automatically be fired. Okay, so it's the minimum stimulus. So when this stimulus is given, only then the membrane potential can rise from resting membrane potential to a particular level which is called as firing level. And from firing level, the action potential will automatically be generated by a positive feedback mechanism. So any stimulus which has intensity less than the threshold stimulus, such stimulus will not be able to produce an action potential, and these stimuli, they are called as sub-threshold stimuli. So such a stimulus which is not able to produce action potential because its intensity is less than threshold stimulus, it's called as sub-threshold stimulus. Another term that we should be aware of is adequate stimulus. So what is adequate stimulus? Let's take an example. We have a receptor, and this receptor, it produces response to when a particular stimulus is given. Let us say in case of this receptor, the stimuli can be more than one. For example, let's take example of rods and cones in our retina. Again, as we all know, these receptors, they are sensitive to light. So once light falls on them, it produces electrical impulses which results in a response. So electrical impulses produce response, that is vision. Okay, action potential is being generated. However, these rods and cones, they can also respond to pressure. So we can easily understand this. If we close our eyes and apply some pressure on our eyes, we are going to see certain spots or white sparks. And this is because these rods and cones start firing when we apply this pressure externally. So here pressure acts as a stimulus. Clearly it means there are two types of stimuli for rods and cones. One is a light stimulus and other is pressure stimulus. However, these rods and cones, they are more sensitive to light stimulus. That means adequate stimulus is the stimulus to which receptor is more sensitive. So in case of rods and cones, this adequate stimulus is the light stimulus. As we have discussed all the basics, now let us go to classification of receptors. There are different receptors in our body, so in order to have better understanding of these receptors, classification becomes necessary. So receptors can be broadly classified as exteroceptors and interoceptors. So what are exteroceptors? Exteroceptors, these are those receptors for which stimulus is being given externally, as the name says, exteroceptors. So stimulus in case of exteroceptors is given externally, whereas in case of interoceptors, stimulus arises from the body parts, arises inside the body, interoceptors. Now, exteroceptors, they can be further divided into different types depending on their location, etc. For example, cutaneous receptors. Cutaneous receptors, they are present in the skin, as the name says. So cutaneous receptors are present in skin. They are type of exteroceptors. Now there are various types of cutaneous receptors. For example, receptors for touch. Now these receptors they include Meissner's corpuscle, Merkel's discs. So these are names of different types of touch receptors. So these are touch receptors. Pressure receptors, they are also present in skin. For example, Pacinian corpuscle. Now, Pacinian corpuscle is the pressure receptor which is present in skin. This is a very important receptor. Another type of receptor present in skin, it's the receptor for cold. Receptor for cold is the Krause and bulb. Krause and bulb is receptor for cold. That means it's a temperature receptor. Similarly, we have another receptor for warmth, and the receptor for warmth is Ruffini nerve endings. Ruffini nerve endings act as receptor. So Krause and bulb and Ruffini nerve endings, they are receptors for temperature. Similarly, we have receptors for pain, which are called as nociceptors. So all these receptors, they are cutaneous receptors, and the stimulus is given externally, so they are included in exteroceptors. Another class of exteroceptors is called as chemoreceptors. So as the name says, the stimulus is chemical in nature in case of chemoreceptors. These receptors are further of two types, that's receptors for taste and receptors for smell. As we can understand, in both these cases the stimulus is chemical. So receptors for taste, they are nothing but taste buds, and receptors for smell, these receptors are olfactory receptors. So these are examples of chemoreceptors. Third class of receptors in exteroceptors is the teloreceptors. So if we try to understand the meaning of this term teloreceptors, it means these are receptors for which stimulus is produced at some distance. So which sensations can be perceived when the stimulus is produced at some distance? Obviously hearing and vision. So vision, that means rods and cones are teloreceptors in this case, and for hearing we have hair cells which are receptors. So rods and cones and hair cells are examples of teloreceptors. Now, interoceptors, these are the receptors for which stimulus arises inside the body. Interoceptors can be divided into two classes. These are visceroreceptors and proprioceptors. So what are visceroreceptors? Visceroreceptors, as the name says, stimulus for these receptors arises in viscera. So these receptors include receptors such as baroreceptors, osmoreceptors, baroreceptors, stress receptors, osmoreceptors, they are all included in visceroreceptors. Proprioceptors, on the other hand, they are stimulated when there is change in the position of body parts. These include receptors such as Golgi tendon organ and muscle spindle. So proprioceptors, the stimulus for these arises whenever there is change in the position of body parts. Golgi tendon organ and muscle spindle are examples of proprioceptors. So these are different classes of receptors that we should be aware of. Now let us discuss another important aspect of receptors, that is properties of receptors. There are certain properties of receptors which are very important and which we should definitely know, and these are also important for exams. So first important property of receptors is it's called as the specificity of response. So what do we mean by specificity of response? It means the pathway from receptor to its center in the brain. If this pathway is stimulated anywhere, the response produced will always be same. This specificity of response, this is also called as Muller's doctrine of specific nerve energies. Muller's doctrine of specific nerve energies, it's one of the properties of receptors. Another important property of receptors is called as adaptation. This is a very important property of receptors. Adaptation means, let us say this is a receptor and we apply stimulus to it continuously for some time. So when this stimulus is applied continuously, after some time there will be decrease in the discharge from this receptor. That means decrease in the discharge of impulses from this receptor. In other words, there will be desensitization of receptor. So this process of desensitization is called as adaptation of receptor. Adaptation of receptor. Now let us say whenever we wear clothes, when we wear new clothes, initially we are able to feel the touch of the clothes, but after some time the receptors get undergo adaptation and then we do not feel clothes on our skin. So this is a classical example of adaptation of receptors. Now depending on the time taken for adaptation, receptors can be divided into two types. So depending upon adaptation time, we have two types of receptors. So one class is called as phasic receptors, and other type of receptors, they are tonic receptors. Phasic receptors, these are fast adapting receptors. Phasic receptors are fast adapting receptors, and tonic receptors on the other hand, these are slowly adapting receptors. Slowly adapting receptors. Phasic receptors, they anatomically these are encapsulated nerve endings. Phasic receptors are encapsulated nerve endings, whereas tonic receptors, these are free or expanded nerve endings. Tonic receptors are free or expanded nerve endings. Phasic receptors, they can determine rate of change of stimulus with respect to time. Rate of change of stimulus with respect to time. And tonic receptors, they can only detect a steady stimulus. Examples of phasic receptors include Pacinian corpuscle and hair cells. So Pacinian corpuscle, they are rapidly adapting receptors. Similarly, hair cells in the inner ear, these are also rapidly adapting receptors, hence phasic receptors. Tonic receptors, examples of tonic receptors include receptors such as muscle spindle and receptors for cold, which are present in the skin. So muscle spindle, cold receptors, these are examples of tonic receptors. So now let us try to understand this with the help of a simple diagram in the form of a graph. Okay, let's draw a graph and we'll take Pacinian corpuscle and muscle spindle. So this on y-axis we have the impulses generated, and on x-axis we show the time. Now when stimulus is continuously given, in case of Pacinian corpuscle, adaptation will take place rapidly. That means impulses will reach to zero in this much time, which is very less. On the other hand, in case of muscle spindle, again when the stimulus is given continuously, the impulses generated will start decreasing, but they will reach to level 0 in much greater time. So these are slowly adapting receptors, muscle spindle. So this is all about the adaptation of receptors. Now another important property of receptors, it's called as Weber-Feschner law. So what is Weber-Feschner law? This law can be stated in the form of an equation. So R is directly proportional to log of S, which implies R is equal to K into log of S. In this equation, R is the response which is produced after stimulus is given, and K is the strength of the stimulus that we apply to the receptor. K is the constant. So from this equation, we can understand that if response has to be doubled, then the strength of stimulus has to be increased almost 100 times. So when the stimulus is strength of stimulus is increased 100 times, only then response will be doubled. So this law is called as Weber-Feschner law. Now let us discuss the last property of receptors. This is called as receptor potential. So receptors, once they are stimulated, they are able to generate a transmembrane potential difference, which is called as receptor potential. So let us say this is a receptor and we give stimulus to this receptor. This results in generation of a transmembrane potential difference in the receptor, which persists for a very short duration of time, and this potential is called as receptor potential. And eventually this receptor potential will result in generation of action potential in the sensory nerve. So we will understand this whole mechanism by taking example of Pacinian corpuscle. So before that, let's write some properties of receptor potential. So first important property is receptor potential is a transmembrane potential. Second important property is receptor potential is it's produced when stimulus is given to the receptor and it is non-propagating in nature. It's non-propagating in nature. Another important property is that receptor potential is short-lived, therefore it's called as transient receptor potential. Receptor potential is short-lived, hence called as transient receptor potential. Now, another important property of receptor potential is this receptor potential is very much similar to excitatory postsynaptic potential. Excitatory postsynaptic potential will be discussed in case of synapse. Okay, now this receptor potential, it results in generation of action potential, therefore receptor potential is also called as generator potential. Why is it called as generator potential? Because it results in generation of action potential in the sensory nerve. And the last property of receptor potential is that receptor potential does not follow all or none law. All or none law is not followed in case of receptor potential. Now, without wasting any time, let's move to the mechanism of generation of receptor potential by taking example of Pacinian corpuscle. So let us try to understand Pacinian corpuscle first. So we have a central core fiber. So this is the central core, and here these are the Schwann cells. So this fiber is myelinated, and in between these Schwann cells, these are the nodes of Ranvier. So this node is the first node of Ranvier. First node of Ranvier is important in the generation of action potential in this nerve. So we have the central core node of Ranvier, and this whole thing is the sensory nerve. Now Pacinian corpuscle will be present right at the periphery, and it's present in the form of certain lamellae. So these lamellae, these are parts of this receptor. So there are various lamellae present in the Pacinian corpuscle. So this receptor is, so these are lamellae, and this receptor is called as Pacinian corpuscle. Pacinian corpuscle is a receptor present in the skin, and it senses the pressure. So it's a receptor for pressure present in the skin. Pacinian corpuscle, pressure receptor. Now let us try to understand how action potential, how receptor potential will be generated here, and how that will result in generation of action potential. So once the stimulus is applied in the form of pressure to these lamellae, it will result in change in shape of the Pacinian corpuscle. So initially this pressure applied to Pacinian corpuscle, so pressure stimulus is given. This pressure stimulus will be propagated along these lamellae, and it will cause compression of the lamellae. So there will be compression of lamellae, and eventually these waves of compression will reach to the central core, and this will result, so there will be change in shape of Pacinian corpuscle, and that will result eventually in disruption of central core. So there will be deformation of central core. Once in the central core, mechanical sodium channels are present. So once there will be deformation is a mechanical change. So these mechanical sodium channels will start opening, and once these mechanical sodium channels will open, that will result in influx of sodium, and this influx of sodium will result in generation of an will result in generation of a small potential difference, which is called as the receptor potential. This influx of sodium will result in generation of receptor potential. So, thus the receptor potential is generated in the Pacinian corpuscle after pressure stimulus is given. Now, how will this receptor potential cause generation of action potential in the nerve? So, this receptor potential will form certain circuits of currents. So, these will be local circuits of currents, and it will reach till the first node of Ranvier. So, this receptor potential, it will be propagated to the first node of Ranvier by forming local circuits of currents. It will not be propagated as the action potential propagates in a nerve fiber. It will be propagated by local circuits of currents, and once it reaches first node of Ranvier, in the first node of Ranvier, voltage gated sodium channels will be present, and which will open as this potential is received, as this voltage is received, and that will result in generation of action potential, which will be propagated from one node of Ranvier to another in the sensory nerve. So action potential will be generated in this nerve. So that was all about the receptors. Thank you.